Good evening, guys. Whether my voice is audible? Yes. Is it audible? Fatima, Mahalakshmi, Apeksha, and Shweta. Okay. So let us wait for 4 more minutes. Then we will start the class. As I'm waiting for other students to enter in, let them come and I'm going to start the session. Yes. So most of you are attending first time. Some of you attended the early sessions, and you know that in biology class you must switch on your video. So I'm giving you 4 minutes time so that you can settle down at one place where you can keep your mobile and switch on the video. Whenever I ask you the question personally, I request you to switch on the mic and answer. If you don't know also, you can say, no, ma'am, I don't know. Like this you can say, and don't hesitate whether you are from Kannada medium or English medium or any other medium. Please do respond and interactive. Then only you will understand much better and you can utilize the sessions. Otherwise, it will become simply like a repetition from the college. That's it. Make use of the sessions. And as I am saying, from the first class, your video should be switched on. When I switch on my video, I want to see the faces of every student. So I request you all, I instruct you all to switch on the video once the session starts. At 6:05 p.m., exactly at 6:05 p.m., I'm going to start the session. So please sit in a comfortable place and a comfortable position so that you can keep your mobile correctly and keep your book correctly, and you can write the notes whenever required. Yes. Yeah, I hope, I think all of you are ready with your arrangements. Like, yeah, okay. Sit in a comfortable position and please switch on your video once the class starts. Don't make me to repeat the things while class is going on. While class is going on, so I should not repeat, only to switch on the video like a small children. Okay. Okay, 9 students. Komala's audio is not connecting. Fine. Students are here. Okay. Fine. Good. In the last session, we discussed regarding the sexual reproduction in plants. In that, we were learning regarding the concept of pole and pistil interaction. So last class episode was pole and pistil interaction, guys. Today, we are learning regarding the next episode, that is a fertilization. So what is a recap about the last session? The recap about the last session is pollen pistil interaction, where pollen is landed on the stigma, pollination happened, right? Okay. Now let us begin with the session. Okay, 11 students are here. Let us begin. Just a minute. I hope the screen is visible to everyone. Yeah, I request everyone to switch on the video so that we can start the session. Yes. Yes, guys, please switch on your videos. Fatima, Mahalakshmi, Apeksha, Jyoti, Priyanka, switch on the video. Thank you. Manya, Shweta, switch on the video. Thank you. Komala, Netravati, Diksha. Okay. Fatima, Mahalakshmi, Apeksha, switch on your videos. Don't make me to repeat it whole session. Fine. So the last episode was a pole and pistol interaction. Now the girl side and boy side parents agreed for the marriage. Now marriage is going to happen. This is not about the serial. This is regarding the sexual reproduction in plants. So today we are going to learn regarding the fertilization. So what do you mean by fertilization? Can anyone define in your own words? What do you mean by fertilization? Yeah. You can just tap on a screen. Your mic will be switched on so that you can tell the answer. Fatima, switch on the video. Thank you, Fatima. Yeah. Heli, fertilization andre enu? So pollination aitu. Somehow boy impressed the girl. Pollen pistil interaction got over. Now we have the fertilization. So fertilization is nothing but a fusion of gametes. Gametes are nothing but the sex cells, guys. So we know that in angiosperms, so in angiosperms plant alli enide, male gamete is motile. Motile andre enu? Able to move anta. Whereas female gamete is non-motile. Non-motile means what? It will not move. It will be in the embryo sac. So the fusion of male gamete with the female gamete is called as fertilization. ಇದನ್ನ ಯಾರು ಕಂಡು ಹಿಡಿದ್ರು? ಸ್ಟ್ರಾಸ್ಬರ್ಗರ್ ಅನ್ನೋರು ಮೋನೋಟ್ರೆಪಾ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ 1884 ಅಲ್ಲಿ ಕಂಡು ಹಿಡಿದ್ರು. See, 1884 he discovered, and now we are facing the issue because we want to write exam on it, ಅಲ್ವಾ? So ಸ್ಟ್ರಾಸ್ಬರ್ಗರ್ ನ ಆಮೇಲೆ ಬೈಕೊಳ್ಳೋಣ. This process of fertilization is completed in the 4 steps. So fertilization is not that male gamete is fused with female gamete ಆಗೋಯ್ತು ಅಂತ ಅಲ್ಲ. It is based on certain factors. ಅದಕ್ಕೂ ತನ್ನದೇ ಆದಂತಹ certain conditions ಇದೆ. ಸೊ ಈಗ ಮ್ಯಾರೇಜ್ಗೆ ಅಪ್ಪ ಅಮ್ಮ ಈ ಕಡೆ ಅಪ್ಪ ಅಮ್ಮನೂ ಒಪ್ಪಿದ್ದಾರೆ, ಈ ಕಡೆ ಅಪ್ಪ ಅಮ್ಮನೂ ಒಪ್ಪಿದ್ದಾರೆ. ಬಟ್ ಬೇಸ್ಡ್ ಆನ್ ಸರ್ಟನ್ ಟರ್ಮ್ಸ್ ಅಂಡ್ ಕಂಡೀಷನ್. ನಮ್ ಹುಡುಗಿ ನಾನು ಹೀಗೆ ನೋಡ್ಕೋಬೇಕು, ನಮ್ ಹುಡುಗಿ ನಾನು ಬೈಕಲ್ಲೇ ಕರ್ಕೊಂಡು ಹೋಗ್ಬೇಕು, ನಮ್ ಹುಡುಗಿ ಬಸ್ ಹತ್ತಲ್ಲ, ಹೀಗೆ ಟರ್ಮ್ಸ್ ಅಂಡ್ ಕಂಡೀಷನ್ಸ್ ಇರತ್ತಲ್ವಾ. ಅದೇ ರೀತಿ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗುವಾಗ there is a certain terms and conditions. So what is that terms and condition? Let us see. Germination of pollen grain and pollen tube. So pollen grain stigma ಮೇಲೆ ಹೋಗಿ ಕುತ್ಕೊಳ್ಳೋದಷ್ಟೇ ಅಲ್ಲ, pollen grain has to germinate and pollen and produce pollen tube. ಸೊ ಇಲ್ಲಿ ಏನ್ ಮಾಡ್ಬೇಕು? ಹುಡುಗ ಜಸ್ಟ್ ಅಪ್ಪ ಅಮ್ಮನ ಕನ್ವಿನ್ಸ್ ಮಾಡೋದಲ್ಲ, ಆ ಹುಡುಗಿ ಅಪ್ಪ ಅಮ್ಮನ ಕನ್ವಿನ್ಸ್ ಮಾಡೋದಲ್ಲ, ಅವ್ರ ಅಪ್ಪ ಅಮ್ಮನ ಕನ್ವಿನ್ಸ್ ಮಾಡಿ ಅದೇ ರೀತಿ ನಡ್ಕೋತಾನೋ ಇಲ್ಲೋ ಎಂಗೇಜ್ಮೆಂಟ್ ಇಂದ ಮ್ಯಾರೇಜ್ ವರ್ಗು. ಸೊ ದಟ್ ಆಲ್ಸೋ ಮ್ಯಾಟರ್ಸ್, ರೈಟ್? ಅಥವಾ ಎಂಗೇಜ್ಮೆಂಟ್ ಅಷ್ಟೇ ಆಗಿ ಎಂಗೇಜ್ಮೆಂಟ್ ಬ್ರೇಕ್ ಆದ್ರೆ? That is why germination of pollen grain and pollen tube is necessary. So pollen, when it reaches the stigma, it is necessary to germinate and reach the pollen and produce the pollen tube. Please switch on your videos, all the students. Yeah. Entry of pollen tube into the ovule. Don't think that ನಮ್ಮ ಮನೆ ಬ್ಯಾಕ್ ಗ್ರೌಂಡ್ ಸರಿ ಇಲ್ಲ, ನನ್ನ ವಾಲ್ ಪ್ಲೇನ್ ಇಲ್ಲ, ಪಕ್ಕದಲ್ಲೇ ನಾನು ಐ ಕ್ರಿಯೇಟೆಡ್ ಅ ಮೆಸ್. Don't worry, we will not see that. We want to see only your face. Okay. Entry of pollen tube into embryo sac. First, entry of pollen tube into the ovule. ಒಳಗಡೆ pollen tube ಹೋಗ್ಬೇಕು, ovule ಒಳಗಡೆ. Next, entry of pollen tube into a embryo sac. Ovule ಒಳಗಡೆ ಹೋಗಿ it has to reach the embryo sac. Embryo sac ಅಲ್ಲಿ ಏನಿದೆ? ಹುಡುಗಿ. Embryo sac ಅಲ್ಲಿ ಇರೋದು egg, right? Don't write ಹುಡುಗಿ. Entry of pollen tube into embryo sac. Next, after that, there will be a fusion of gametes. ಗಟ್ಟಿ ಮೇಳ ಆಗತ್ತೆ. So these all are the terms and conditions for the fertilization. ಇಲ್ಲಿ ಏನೇನ ಗಮನಿಸಬೇಕು? We have to concentrate on 3 points, guys. ಒಂದನೇದು definition, which is important for our PUC exam. The fusion of male gamete with the female gamete. ಎರಡನೇದು ಯಾರು ಕಂಡು ಹಿಡಿದ್ರು? It was discovered by Strasburger in a monotrapa plant. So it is a CET and a NEET question. Then fertilization happens is completed based on 4 terms and conditions or 4 steps. Yeah, steps. So these steps can be asked in CET also. These steps can be asked in NEET also, as well as PUC board exam also. Hege anta helidre NEET alli hege keltare, they will give the what we can say serial wise list. First ಏನ್ ಆಗಬೇಕು, germination of pollen grain and pollen tube. That is the first step. Entry of pollen tube into the ovule. Second, entry of pollen tube into a embryo sac. Third step, fusion of gamete is a fourth step. So ಇದನ್ನ ಅವರು ಜಂಬಲ್ ಮಾಡ್ಕೊಡ್ತಾರೆ. Then they will ask, tell the exact or serial steps ಅಂತ. That is a NEET question. Or sometimes when a CET exam is tough, that will be a CET questions too. And for 2 marks, they will ask in a PU board exam regarding steps to complete the fertilization. I hope you understood the brief concept of fertilization. Did you get, guys? Yeah. Okay. Please note down for 2 points. Yeah. Fusion of male and gamete with female gamete. First point barkoli. Second point barkoli discovery bagge. And write steps in fertilization anta barudu. Write all the steps. Okay. Manya, switch on the video. Diksha DM, Netravati. Every class al nan helak agalla. Mahalakshmi, Jyoti, Komala. Please switch on your videos. Apeksha M. Write it fast. I told you how to write during the session. Just scribble it on. Don't do a separate do separate book for your recognition, but don't do on decorating your handwriting. Just scribble it so that we can move to the other concepts fast. Fine. Netravati, switch on the video. Manya, Jyoti, finally switch on the video. Thank you. Then Komala. Some are playing hide and seek with me. ನಾನು ಹಿಂಗೆ ನೋಡೋ ಅಷ್ಟೊತ್ತಿಗೆ ವಿಡಿಯೋ ಆಫ್ ಮಾಡ್ಬಿಡ್ತಾರೆ. Find some support to keep the mobile so that you can keep the mobile correctly and you can concentrate on writing. One bottle ಏನಾದರೂ ತಗೊಂಡು ಅದು support ಇಟ್ಕೊಳ್ಳಿ. Some are facing the issue for that. Okay, write it fast, girls. Did you complete? Be fast, please. So sometimes I will be explaining the slide. No, ನಾನು 2 ಸಲ explain ಮಾಡ್ತೇನೆ slide ಅನ್ನ. ನಾನು 2ನೇ ಸಲ explain ಮಾಡುವಾಗ just write it down so that the time will be saved. Shall I proceed with the next slide? Okay, thank you. Fine. Germination of pollen grain. So what is the first step? Germination. How the pollen grain will germinate? After pollination, pollen grain germinate on the stigma of the carpel. So we know na stigma of the carpel. This is a stigma, this is a style, and this is a ovary, right? So pollen grain, when it falls on a stigma, stigma will be having a sticky surface. Pollen grain ಏನಾಗುತ್ತೆ ಅದರ ಮೇಲೆ ಅಂಟ್ಕೊಳ್ಳುತ್ತೆ. ಯಾಕೆಂದ್ರೆ ಸ್ಟಿಗ್ಮಾಗೆ ಸ್ಟಿಕ್ಕಿ ಸರ್ಫೇಸ್ ಇದೆ. pollen grain ಏನ್ ಮಾಡುತ್ತೆ, it absorbs moisture and sugar content from the stigma. ಸ್ಟಿಗ್ಮಾದಲ್ಲಿ ಮಾಯಿಶ್ಚರ್ ಇರುತ್ತೆ, ತೇವಾಂಶ ಮತ್ತೆ sugar content which will be absorbed by the pollen grain. A small pollen grain is or nothing but a microspore ಅಂತ ಏನ್ ಕರಿತೀವಿ, ಅದನ್ನ ಅಬ್ಸಾರ್ಬ್ ಮಾಡ್ಕೊಂಡು ಇಟ್ ವಿಲ್ ಸ್ವೆಲ್ ಅಪ್ ಅಂದ್ರೆ ಊದ್ಕೊಳ್ಳುತ್ತೆ ಅಂತ, ಊದ್ಕೊಳ್ಳುತ್ತೆ. ದ ಮೈಕ್ರೋಸ್ಪೋರ್ ವಿಲ್ ಸ್ವೆಲ್ ಅಪ್, ದೊಡ್ಡದಾಗತ್ತೆ. ದ ಇಂಟೈನ್ ಆಫ್ ಪೋಲನ್ ಗ್ರೇನ್ ಗ್ರೋಸ್ ಔಟ್ ಥ್ರೂ the any one germinal pore of a exine in the form of a tube called pollen tube. So ಏನಾಗುತ್ತೆ, when we see the structure of a pollen grain guys, microspore structure ನೋಡಿದ್ರೆ it is a haploid in nature. Haploid ಅಂದ್ರೆ ಅದರಲ್ಲಿ ಸಿಂಗಲ್ ಸೆಟ್ ಆಫ್ ಕ್ರೋಮೋಸೋಮ್ ಇರುತ್ತೆ. It will be having 2 layers. ಅದಕ್ಕೆ ಎಷ್ಟು ಸೆಲ್ ಲೇಯರ್ಸ್ ಇದೆ? 2 ಲೇಯರ್ ಇದೆ. ಔಟರ್ ಲೇಯರ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಎಕ್ಸೈನ್. ಔಟರ್ ಲೇಯರ್ ಗೆ ತನ್ನದೇ ಆದಂತಹ ಒಂದು ಶೇಪ್ ಇರುತ್ತೆ. ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಎಕ್ಸೈನ್ ಅಂತ ಕರೀತಾರೆ. ಒಳಗಡೆ ಲೇಯರ್ಗೆ ಇಂಟೈನ್ ಅಂತ ಕರೀತಾರೆ. ಸೊ ಈ ಇಂಟೈನ್ ಏನಾಗತ್ತೆ, ವೆನ್ ದ ಸೆಲ್ ಎನ್ಲಾರ್ಜಸ್, ಮೈಕ್ರೋಸ್ಪೋರ್ ಅಥವಾ ಪೋಲನ್ ಗ್ರೇನ್ ದೊಡ್ಡದಾಗ್ತಾ ಆಗ್ತಾ ಈ ಇಂಟೈನ್ ಲೇಯರ್ ಇಟ್ ವಿಲ್ ಬಿ ಸಾಫ್ಟ್. ಸೊ ಇಟ್ ವಿಲ್ ಎಮರ್ಜ್, ಈ ತರ ಟ್ಯೂಬ್ ತರ ಆಗತ್ತೆ. ಟ್ಯೂಬ್ ತರ ಆಗಿ ಅದು ಎಕ್ಸೈನ್ ಲೇಯರ್ ಇಂದ ಏನಾಗತ್ತೆ, ಹೊರಗಡೆ ಬಂದು ಇಟ್ ಫಾರ್ಮ್ಸ್ ಅ ಟ್ಯೂಬ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್. ಸೊ ದಿಸ್ ಟ್ಯೂಬ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ this tube like structure is called as pollen tube. ಇದಕ್ಕೆ pollen tube ಅಂತ ಕರೀತಾರೆ. I hope you understood how pollen tube is formed. Right. So pollen tube ಅಲ್ಲಿ ಏನಿರುತ್ತೆ ಅನ್ನೋದನ್ನ ನೋಡೋಣ. One pollen tube develops in capsula and most of angiosperm is called as monosiphonous type. Monosiphonous condition ಅಂದ್ರೆ ಏನು? ಒಂದು ಪೋಲನ್ ಗ್ರೇನ್ ಇಂದ ಒಂದೇ ಪೋಲನ್ ಟ್ಯೂಬ್ ಬಂದಾಗ that condition is called as monosiphonous. Very important for the competitive exam. Monosiphonous. ಇಷ್ಟು ಉದ್ದ ನಾವು ಬರ್ಕೊಳ್ಳೋ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. One pollen tube from one pollen. One pollen tube from one pollen. Next, in certain families ಅಲ್ಲಿ ಏನಾಗತ್ತೆ, ಮಾಲ್ವೇಸಿಯೆ, ಕುಕ್ಕುರ್ಬಿಟೇಸಿಯೆ, ಕುಕ್ಕುಂಬರ್, ಸೌತೆಕಾಯಿ, ಕುಂಬಳಕಾಯಿ, ಸೊ ಇವೆಲ್ಲ ಬರೋದು ಕುಕ್ಕುರ್ಬಿಟೇಸಿಯೆ ಅಲ್ಲಿ. ಸೊ ಈ ಫ್ಯಾಮಿಲಿಯಲ್ಲಿ ಏನಾಗತ್ತೆ, ಮೆನಿ ಪೋಲನ್ ಟ್ಯೂಬ್ಸ್, ಲೈಕ್ ಇಸ್ ಮೋರ್ ದನ್ ಒನ್ ಪೋಲನ್ ಟ್ಯೂಬ್, ಒಂದಕ್ಕಿಂತ ಹೆಚ್ಚು ಪೋಲನ್ ಟ್ಯೂಬ್ಗಳು ಏನಾಗ್ತವೆ, ಪೋಲನ್ ಗ್ರೇನ್ ಇಂದ ಹೊರಗಡೆ ಬರ್ತವೆ. ಆ ಕಂಡೀಷನ್ಗೆ ಪಾಲಿಸಿಫಾನಸ್ ಕಂಡೀಷನ್ ಅಂತ ಕರೀತಾರೆ. ಮೋನೋ ಸಿಂಗಲ್, ಒನ್ ಪೋಲನ್ ಟ್ಯೂಬ್. ಪಾಲಿ ಮೆನಿ, ಸೊ ಮೋರ್ ದನ್ ಒನ್ ಪೋಲನ್ ಟ್ಯೂಬ್. ದಟ್ಸ್ ಇಟ್. ನೆಕ್ಸ್ಟ್, ಇಂಪಾರ್ಟೆಂಟ್ ಫಾರ್ ಕಾಂಪಿಟೇಟಿವ್, ಇದು ಮೋನೋ ಮತ್ತೆ ಪಾಲಿಸಿಫಾನಸ್ ಕಂಡೀಷನ್. ಓಕೆ, this one and this one. When the pollen tube comes down from the stigma, so stigma ಮೇಲೆ ಏನಾಗಿದೆ ಇಲ್ಲಿ, pollen tube is formed. ಈ pollen tube form ಆಗಿದೆ. Now pollen tube should enter inside the style. Next ಅದ್ರ root ಎಲ್ಲಿ ಹೋಗ್ಬೇಕು, style ಗೆ ಹೋಗ್ಬೇಕು. So when the pollen tube comes down from the stigma into the style, first of vegetative nucleus enter into the style. Sorry, one ನಿಮಿಷ. Yeah. ಸೊ ಫಸ್ಟ್ ಆಫ್ ವೆಜಿಟೇಟಿವ್ ನ್ಯೂಕ್ಲಿಯಸ್. ಸೊ ಪೋಲನ್ ಗ್ರೇನ್ ಅನ್ನ ತಗೊಂಡಾಗ ಅದ್ರಲ್ಲಿ ಒಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ ಅಲ್ವಾ, ಇಟ್ ವಿಲ್ ಬಿ ಅಂಡರ್ ಗೋಯಿಂಗ್ ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್. ಆ ನ್ಯೂಕ್ಲಿಯಸ್ ಏನಾಗ್ತಾ ಇರತ್ತೆ, ಇಟ್ ವಿಲ್ ಬಿ ಅಂಡರ್ ಗೋಯಿಂಗ್ ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್. ಸೊ ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ಆದಾಗ ಪೋಲನ್ ಗ್ರೇನ್ ಅಲ್ಲಿ 2 ನ್ಯೂಕ್ಲಿಯಸ್ ಆಗ್ತವೆ. ಒಂದು ವೆಜಿಟೇಟಿವ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಅಂತ, ಇನ್ನೊಂದು ದ ಅದರ್ ಒನ್ ಇಸ್ ಜನರೇಟಿವ್ ನ್ಯೂಕ್ಲಿಯಸ್. 2 nucleus are formed by the mitotic division of the nucleus in the pollen grain. One is vegetative, the other is generative. So first pollen tube style ಅಲ್ಲಿ ಹೋದಾಗ, ಸ್ಟೈಲ್ ಒಳಗಡೆ ಹೋಗುವಾಗ ಫಸ್ಟ್ ಇರೋದು ವೆಜಿಟೇಟಿವ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ, ಸೆಕೆಂಡ್ ಜನರೇಟಿವ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ. The tube nucleus always occupies in terminal position. Vegetative nucleus controls the growth of pollen tube. Vegetative nucleus ಏನ್ ಮಾಡುತ್ತೆ, it controls the pollen tube. ಸುಮ್ನೆ ಬೆಳಿತಾ ಹೋಗಲ್ಲ pollen tube. It is controlled by the vegetative nucleus. Meanwhile, generative nucleus ಅಥವಾ generative cell ಏನಿದೆ, ಅದರಲ್ಲಿರುವ ನ್ಯೂಕ್ಲಿಯಸ್ ಮೈಟೋಟಿಕಲಿ ಡಿವೈಡ್ ಆಗಿ it forms 2 male gamete. Very important for competitive guys. ಪೋಲನ್ ಗ್ರೇನ್ ಅಲ್ಲಿರುವ ಜನರೇಟಿವ್ ಸೆಲ್, ಪೋಲನ್ ಗ್ರೇನ್ ಅಲ್ಲಿ 2 ಸೆಲ್ಗಳು ಆಗ್ತವೆ. ಒಂದು ಜನರೇಟಿವ್ ಸೆಲ್, ಇನ್ನೊಂದು ವೆಜಿಟೇಟಿವ್ ಸೆಲ್. ದ ಜನರೇಟಿವ್ ಸೆಲ್ ವಿಲ್ ಬಿ ದೇರ್, ನೋ, ಇಟ್ ಡಿವೈಡ್ಸ್ ಮೈಟೋಟಿಕಲಿ ಟು ಫಾರ್ಮ್ 2 male gametes. So 2 male gametes are formed by generative cell, not by vegetative cell. ನೆನಪಲ್ಲಿ ಇಟ್ಕೊಳ್ಳಿ. Boron element and calcium ions are essential for the growth of pollen tube. Very important. ಎಲ್ಲ ಇಂಪಾರ್ಟೆಂಟ್ ಅಂತಾರೆ ಮೇಡಮ್, ತಲೆ ಕೆಟ್ಟಿದೆ ಅಂತ ಹೇಳ್ಬೇಡಿ, because we are discussing regarding the NEET exam. So every word is important here. Boron element and calcium ions ಅಂತ ಏನಿದೆ, so these are the elements which are needed for the growth of pollen tube. Pollen tube healthy ಆಗಿ on time ಆಗಿ grow ಆಗಕ್ಕೆ these elements are necessary. And best temperature for growth of pollen tube will be 20 to 30 degree Celsius ಇರತ್ತೆ. And pollen tube shows apical growth. Apical growth ಅಂದ್ರೆ if this is a pollen tube guys, ಅದು ಹೇಗೆ ಗ್ರೋ ಆಗತ್ತೆ ಅಂದ್ರೆ ಟಿಪ್ ರೀಜನ್ ವಿಲ್ ಬಿ ಗ್ರೋಯಿಂಗ್. ಟಿಪ್ ರೀಜನ್ ಏನಾಗ್ತಿರತ್ತೆ, ಎಪೆಕ್ಸ್ ಅಂತ ಏನ್ ಹೇಳ್ತೀವಿ, ಎಪೆಕ್ಸ್ ಅಂತ ಏನ್ ಹೇಳ್ತೀವಿ, ಸೊ ದಟ್ ವಿಲ್ ಬಿ ಗ್ರೋಯಿಂಗ್. ಜಸ್ಟ್ ಅ ಮಿನಿಟ್. ಎಪೆಕ್ಸ್ ಅಂತ ಏನ್ ಹೇಳ್ತೀವಿ, ದಟ್ ವಿಲ್ ಬಿ ಗ್ರೋಯಿಂಗ್. Right, that is called as apical growth. Chemotropic movement andre, you can ask me if this is a stigma. If this is a stigma, this is a style and this is a ovary, pollen tube will germinate agi hige hege hogatte. There will be no Google map, right? So there is no one to tell you hige hogu anta, ಹೊರಗಡೆ ಹೋಗಬಹುದಾಗಿತ್ತಲ್ವಾ. It is by the chemotropic movement. Chemotropic ಅಂದ್ರೆ chemical synthesis, chemicals ಇಂದ attract ಆಗತ್ತೆ. Stigma will release certain chemicals which makes pollen tube to go down the style. This is called as chemotropic movement, guys. Okay, chemotropic. I hope you understood this concept. ಅರ್ಥ ಆಯ್ತಾ? Okay, ಬರ್ಕೊಂಡ್ರಾ? I mean, most of you are noted or want to note it. Hi, Sahana. I think you logged in late.
Hi, ma'am. Yeah, ma'am.
Yeah. Note it down. If you are completed, let me know. Don't write everything, only write which you feel important. Don't write the whole slide. ನೀಟಿಗೆ ಯಾವ್ದು ಉಪಯೋಗ ಇದೆ ಅದನ್ನ ಬರ್ಕೊಳ್ಳಿ, because this is the neat session. So monocyphanous condition, polycyphanous condition, then boron and calcium ions condition, apical growth and chemotropic. These are the main points related to NEET, guys. Okay. I know everyone are familiar with sending mails. Can you able to send the mails, emails? Mail ಕಳ್ಸಕ್ ಬರತ್ತಾ? ವಾಟ್ಸ್ಆಪ್ ಮೆಸೇಜ್ ಅಲ್ಲ, ಮೇಲ್. ಜಿಮೇಲಿಗೆ ಹೋಗಿ ಮೇಲ್ ಮಾಡಕ್ ಬರತ್ತಾ ಎಲ್ಲಾರ್ಗೂ? Yes or no? Okay, fine. Completed all of you?
Yes, ma'am.
Okay.
Yes, ma'am.
Next, entry of pollen tube into the ovule. So what happens first? Pollen tube enters through the style, then it reaches the ovary. The pollen tube enters in the ovary at the time when ovule becomes mature. Andre yavago enter agalla pollen tube. Pollen tube yavaga enter agatte when the ovule becomes mature. Andre ovule should complete its megagametogenesis. Alli embryo sac formation agirbeku. Avagle pollen tube will enter inside the ovary. Inside the ovary, obturators guides the passage of pollen tube towards the micropyle. Obturators anta ಏನ್ ಹೇಳ್ತೀವಿ, integuments guides the passage of pollen tube. Pollen tube ಗೆ micropyle end ಗೆ ಹೋಗಕ್ಕೆ ಸಹಾಯ ಮಾಡ್ತವೆ. ಯಾವುದು? obturators. ಯಾವ್ದು? ಎದರಲ್ಲಿ? ಓವರಿಯಲ್ಲಿ. Then a mature ovule in which embryo sac also got matured has 3 parts for the entry of pollen tube. ಸೊ ಪೋಲನ್ ಟ್ಯೂಬ್ಗೆ 3 ರೋಡ್ಸ್ ಇರ್ತವೆ. ಇಟ್ ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್ 3 ways to enter the ovule. First way is through the micropyle, the free end of a ovule anta ಏನ್ ಹೇಳ್ತೀವಿ. When pollen tube enters through the micropyle, it is called as pyrogamy. Competitive. Porogamy. Poro ಅಂದ್ರೆ ಪೋರ್ ಎಲ್ಲಿರುತ್ತೆ ಓವ್ಯೂಲ್ ಅಲ್ಲಿ? ಮೈಕ್ರೋಪೈಲ್ ಎಂಡ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ಓಪನ್ ಎಂಡೇ ಅದು ಅಲ್ವಾ? ಓವ್ಯೂಲ್ ಇಸ್ ಕ್ಲೋಸ್ಡ್. ಸೊ ದ ಓಪನಿಂಗ್ ಆಫ್ ovule is nothing but that pore micropyle. So when pollen tube enters through the micropyle, it is called porogamy. How to write this? Porogamy anta barkoli. Entry through the micropyle. Yar entry? Yes entry alla. Pollen tube entry through the micropyle is called as porogamy. Alva? Next. Porogamy, pollen tube entry through the micropyle. Poro, entry through micropyle. It should be recorded in your brain. ಯಾವುದೋ ಒಂದು PUC ಎಕ್ಸಾಮು, ನೀಟ್ ಎಕ್ಸಾಮು ಬರುವಾಗ ಯಾವತ್ತೋ ಒಂದಿನ ಮ್ಯಾಮ್ ಕೊರಿತಾ ಇದ್ವಲ್ಲ, ಇದೇ ಆನ್ಸರ್ ಇರಬೇಕು ಅಂತ ಟಿಕ್ ಹಾಕ್ತೀರಾ ಅಲ್ವಾ? ಸೊ ದ ನೆಕ್ಸ್ಟ್ ಎಂಡ್ ಈಸ್ chalazogamy. In this method, the pollen tube enters into the ovule through the chalaza. So micropyle opposite. This is a micropyle ಅಂದ್ರೆ ಇದರ ಅಪೋಸಿಟ್ ಯಾವ್ದಿರುತ್ತೆ? ಕೆಲಾಜಾ or chalaza. ಕೆಲಾಜಾ ಅಂತಾನೂ ಕರೀತಾರೆ, ಚಲಾಜಾ ಅಂತಾನೂ ಕರೀತಾರೆ. ಸೊ ವೆನ್ ಪೋಲನ್ ಟ್ಯೂಬ್ ಎಂಟರ್ಸ್ ಥ್ರೂ ದ ಚಲಾಜಾ, ಸೊ ಅದಕ್ಕೆ ನಾವು ಚಲಾಜೋಗ್ಯಾಮಿ ಅಂತ ಹೇಳ್ತೀವಿ. ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಚಲಾಜೋಗ್ಯಾಮಿ. ಎಂಟ್ರಿ ಥ್ರೂ ಚಲಾಜಾ, ಚಲಾಜೋಗ್ಯಾಮಿ. ವೆರಿ ಈಸಿ. ಎಂಟ್ರಿ ಥ್ರೂ ಮೈಕ್ರೋಪೈಲ್, porogamy. Entry through chalaza, chalazogamy. Next. Okay. Completed all of you? Next we have chalazogamy, pollen tube entry through chalaza. Pollen tube entry through chalaza. Next we have mesogamy. When pollen tube enters through the integuments, when pollen tube enters through the layers of the ovule. The layers of the ovule are nothing but integuments. So pollen tube yavaga integuments through insert agatto, that is called as mesogamy. Poro, micropyle, chalaza, chalazogamy, chalazo, chalaza, meso, integuments. I repeat, poro, micropyle, chalazo, chalaza, next meso, integuments. Right? So pollen tube integuments through enter adaga, it is called as mesogamy. Did you get, girls? Artha aita? Diagrams are very important. So you can just write there. Refer diagram from textbook ಅಂತ ಎಕ್ಸಾಮ್ ಅಲ್ಲೂ ಹಾಗೆ ಬರಿಬೇಕು ಅಂತ ಅಲ್ಲ, ನೀವು ರೆಫರ್ ಮಾಡಿ ಎಕ್ಸಾಮ್ ಗಿಂತ ಮುಂಚೆ. ದೆನ್ ಡ್ರಾ, ಪ್ರಾಕ್ಟಿಸ್ ಟು ಡ್ರಾ. ಓಕೆ, ಮಹಾಲಕ್ಷ್ಮಿ, ಕೋಮಲ ಡಿಡ್ ನಾಟ್ ಸ್ವಿಚ್ ಆನ್ ದ ವಿಡಿಯೋ ಯೆಟ್. ಜ್ಯೋತಿ, ಫೈನ್. ಲೆಟ್ ಮೀ ಪ್ರೊಸೀಡ್. Okay, let us watch this video. Just a minute. So video play aguvaga, I will be teaching simultaneously. So please do respond to the questions also. Please do watch the video, observe the video. Not watching the video, you have to observe. Understand? Yeah, let us begin. Do concentrate, all of you. Switch on your video. Yes. So this is a fertilization. You are seeing the ovules, embryo sac. You are seeing. Yes. This is a pollination, guys. Okay, pollen grain is landing on the stigma. Now pollen tube has to be formed. See, the pollen tube is formed. In the pollen tube, you are seeing 2 cells. First 2 cells ಕಾಣಿಸ್ತು, vegetative ಮತ್ತೆ generative. Generative cell ಏನ್ ಮಾಡ್ತು, divide ಆಗಿ, when you see, correct ಆಗಿ ನೋಡ್ಕೊಳ್ಳಿ. ನಾನು ಮತ್ತೆ I will replay it, observe it carefully. Pollen tube forms and it emerges. ಎರಡು ನ್ಯೂಕ್ಲಿಯಸ್ ಇದೆ. ಜನರೇಟಿವ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಏನಾಗುತ್ತೆ, ಇಟ್ ಡಿವೈಡ್ಸ್ ಮೈಟೋಟಿಕಲಿ ಅಂಡ್ ಯು ಸಾ ಟೂ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್. ಆ ಬ್ಲೂ ಕಲರ್ ಏನಿದೆ ಅಲ್ವಾ, ದಟ್ ಈಸ್ ಅ ಡಿವಿಷನ್ ಆಫ್ ಜನರೇಟಿವ್ ನ್ಯೂಕ್ಲಿಯಸ್. ಏನ್ ಫಾರ್ಮ್ ಆಗಿದೆ, ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಆರ್ ಫಾರ್ಮ್ಡ್. ಇಲ್ಲಿವರೆಗೂ ಅರ್ಥ ಆಯ್ತಾ ಎಲ್ಲರಿಗೂ? ಎಸ್, ಲೆಟ್ ಅಸ್ ಪ್ರೊಸೀಡ್. ಓಕೆ, ಫೈನ್. ನೆಕ್ಸ್ಟ್ ಈಗ ಯಾ, ಸೊ just so here see, two male gametes entered into the egg apparatus or embryo sac. Idenidu embryo sac alva. So one the male gamete ಏನ್ ಮಾಡ್ತು, it got fused with the egg. ಇಲ್ಲಿ egg ಇರತ್ತೆ. These are the synergids. These are the synergids. Violet color nucleus ಇದೆ ಅಲ್ವಾ, these are the synergids. Yeah, see, this is a synergid, this is a synergid where I am marking. The light sky blue color is a male gamete which fused with the egg, Next we have another male gamete el hoitu, central cell ig hoitu. So in the central cell we will be having two polar nuclei. How many nuclei? Two polar nuclei, right? Yeah, two polar nuclei. Two polar nuclei jote male gamete to fuse agi means what all happened there. One nimisha. N is a male gamete. N is a male gamete. Two polar nuclei ide which has NN chromosome. So it got fused with these two polar nuclei. N plus N plus N. So triploid nucleus is formed at the center. Centrally triploid nucleus. So ಇಲ್ಲಿ ಡಿಪ್ಲಾಯ್ಡ್ zygote ಫಾರ್ಮ್ ಆಯ್ತು. ಅರ್ಥ ಆಯ್ತಾ ಎಲ್ಲರಿಗೂ? Did you get? Okay. Now let us begin. Okay, watch it. Observe the video carefully. Sigma is landing. Pollen tube is formed. Generative nucleus divided to form two male gametes. Two male gametes are entering the embryo sac. One male gamete fused with egg, other male gamete fused with polar nuclei. Now, this is called syngamy and syngamy and triple fusion. Triple fusion ಯಾಕೆ ಅಂತೀವಿ ಅಂತ ಅಂದ್ರೆ 3n ಫಾರ್ಮ್ ಆಯ್ತಲ್ವಾ, ಅಲ್ಲಿ ಟ್ರಿಪ್ಲಾಯ್ಡ್ ಫಾರ್ಮ್ ಆಯ್ತು ಅಲ್ವಾ. See, pollen tube is emerging. Again they are showing generative nucleus, so vegetative nucleus. It divides to form male gametes. Vegetative nucleus, it degenerate ಆಗುತ್ತೆ. ಮೈಕ್ರೋಪೈಲ್ ಇಂಟೆಗ್ಯುಮೆಂಟ್ ಕೆಲಸ. ಮೈಕ್ರೋಪೈಲ್ ಇಂದ ಆದ್ರೆ ಪೋರೋಗ್ಯಾಮಿ, ಕೆಲಸ ಇಂದ ಆದ್ರೆ ಕೆಲಸೋಗ್ಯಾಮಿ, ಇಂಟೆಗ್ಯುಮೆಂಟ್ಸ್ ಇಂದ ಆದ್ರೆ ಮೀಸೋಗ್ಯಾಮಿ. ಎಲ್ಲಿ ಎಮರ್ಜ್ ಆಗತ್ತೆ ಅದ್ರ ಮೇಲೆ. ಸಿ ಹಿಯರ್ ಸಿನರ್ಜಿಡ್ ಇದೆ. ಸಿನರ್ಜಿಡ್ ಏನಾಗುತ್ತೆ, ಡಿಜನರೇಟ್ ಆಗತ್ತೆ. ಸೊ ದಟ್ ದ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ವಿಲ್ ಫ್ಯೂಸ್ ವಿತ್ ದ ಎಗ್. ದ ಅದರ್ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ವಿಲ್ ಗೋ ಟು ದ ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ. ಯು ಕ್ಯಾನ್ ಸಿ ಇಟ್ ನೌ. ಡಿಪ್ಲಾಯ್ಡ್ zygote ಫಾರ್ಮ್ ಆಗಿದೆ. ಒನ್ಸ್ ದ ಎಗ್ ಗಾಟ್ ಫ್ಯೂಸ್ಡ್ ವಿತ್ ದ this one, the other male gamete generate. ಇದಕ್ಕೆ ಡಿಪ್ಲಾಯ್ಡ್ zygote ಫಾರ್ಮ್ ಆದಾಗ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ syngamy, ಫರ್ಟಿಲೈಸೇಷನ್ ಅಂತ ಕರೀತಾರೆ. ನೆಕ್ಸ್ಟ್ ಏನಾಗತ್ತೆ, ಓವರಿ, ಸ್ಟೈಲು, ಸ್ಟಿಗ್ಮಾ ಎಲ್ಲ ಡ್ರೈ ಆಗತ್ತೆ, ವಿದರ್ ಆಫ್ ಆಗತ್ತೆ. ದೆನ್ ಆಫ್ಟರ್ ದಟ್ ಯು ವಿಲ್ ಗೆಟ್ ದ ಫ್ರೂಟ್. ಫ್ರೂಟ್ ಅಲ್ಲಿ ಫ್ರೂಟ್ ಅಲ್ಲಿ ಸೀಡ್ ಏನಾಗಿರುತ್ತೆ, ಓವ್ಯೂಲ್ ಆಗಿರುತ್ತೆ. ಓವ್ಯೂಲ್ಸ್ ವಿಲ್ ಬಿ ಕನ್ವರ್ಟೆಡ್ ಇಂಟು ಸೀಡ್. ದೆನ್ ಓವರಿ ವಿಲ್ ಬಿ ಕನ್ವರ್ಟೆಡ್ ಇಂಟು fruit. Yes, I hope you got it, guys. Whether the video was useful. Now, Iga, all should respond to me. First, what we saw in the video. What is the first step in the video?
Pollen tube formation.
Pollen tube formation. Just a minute. Pollen tube formation ಆಯ್ತು. Next, what we learned? Pollen tube formation ಆದ್ಮೇಲೆ what is the next step? Pollen tube enters. Tell me, Priyanka.
Generative nucleus breakdown.
Okay, generative nucleus breakdown ಅಂತ ಉಪಯೋಗಿಸಬಾರ್ದು ಇಲ್ಲಿ ವರ್ಡ್. ಗುಡ್ ಟ್ರೈ. Generative nucleus divides. Its nucleus undergo division, mitotic division ಆಗಿ ಅಲ್ಲೇನಾಗತ್ತೆ? Generative nucleus forms what? It forms male gametes. Right? Good. Generative nucleus forms male gametes form ಆಗತ್ತೆ. Male gametes form ಆದ್ಮೇಲೆ what is the next step?
One gamete will enter into egg and another will enter into polar nuclei.
Okay, very good, Jyoti. So one gamete ಏನಾಗತ್ತೆ? Egg ಜೊತೆ fuse ಆಗತ್ತೆ. The other gamete, the other one, second one ಏನಾಗತ್ತೆ? It fuses with polar nuclei. So polar nuclei ಎರಡು two polar nuclei ಇರೋದ್ರಿಂದ this is called triploid fusion. So ಇಲ್ಲಿ n fuse ಆಗಿ ಡಿಪ್ಲಾಯ್ಡ್ zygote ಆಗೋದ್ರಿಂದ ಎರಡು ಸಲ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗತ್ತೆ. ಒಂದು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ವಿಲ್ ಫ್ಯೂಸ್ ವಿತ್ ದ ಎಗ್, ದ ಅದರ್ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ವಿಲ್ ಫ್ಯೂಸ್ ವಿತ್ ದ ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ. ದಟ್ ಈಸ್ ವೈ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಡಬಲ್ ಫರ್ಟಿಲೈಸೇಷನ್. ಟ್ರಿಪಲ್ ಫ್ಯೂಷನ್ ಅಂದ್ರೆ ಒಂದು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಎರಡು ನ್ಯೂಕ್ಲಿಯೈ ಜೊತೆ ಫ್ಯೂಸ್ ಆಗೋದ್ರಿಂದ ಟ್ರಿಪಲ್ ಆಗತ್ತೆ. ವಿ ವಿಲ್ ಗೆಟ್ 3n ಟ್ರಿಪ್ಲಾಯ್ಡ್ ಅಲ್ವಾ? ಸೊ ದಟ್ ಈಸ್ ವೈ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಟ್ರಿಪ್ಲಾಯ್ಡ್ ಫ್ಯೂಷನ್. ಡಬಲ್ ಫರ್ಟಿಲೈಸೇಷನ್, ಡಿಪ್ಲಾಯ್ಡ್ ಫ್ಯೂಷನ್. ಅಂಡರ್ಸ್ಟುಡ್ ಆಲ್ ಆಫ್ ಯು? ಅರ್ಥ ಆಯ್ತಾ? ಫೈನ್. ಎಂ ಹರ್ಷಿತಾ, Suma, both of you switch on the video, please. Now, entry of pollen tube. This all we learned through the video. Fusion of gametes occur. So fusion of gamete ಆದ್ಮೇಲೆ the second male gamete which fuses with the diploid secondary nucleus or to the polar nuclei, it forms into triploid ಅಂತ ಏನ್ ಹೇಳಿದ್ನಲ್ಲ, triploid ಏನಾಗತ್ತೆ? ಅದು triploid cell. It forms into triploid cell. ಆ ಸೆಲ್ಲಿಗೆ we will call it as primary endosperm nucleus. The triploid cell is considered as primary endosperm nucleus. Then a zygote is formed by true fertilization and develops into an embryo. Then triploid primary endosperm nucleus ಏನ್ ಮಾಡತ್ತೆ? It provides nutrition to the embryo. It provides nutrition to the embryo. This is regarding the double fertilization and triple fusion. Illu certain conditions ಇದೆ. The fertilization in which non-motile gametes are carried to female gamete through pollen tube, it is called stephanogamy. So fertilization ಅಲ್ಲಿ when non-motile gametes ಅಂದ್ರೆ male gametes ಅಥವಾ sperms non-motile ಇದ್ರೆ, if they did not have tail, then it is called as stephanogamy ಅಂತ ಹೇಳ್ತಾರೆ. Then entry of more than one pollen tube into the ovule. ಒಂದಕ್ಕಿಂತ ಹೆಚ್ಚು pollen tube ovule ಅಲ್ಲಿ ಎಂಟ್ರಿ ಆದಾಗ it deals with the polyspermy, many sperms, poly, many, spermy, sperms. This condition is called polyspermy. Important for competitive exam, please note it down. The fertilization in which non-motile gametes are carried to female gamete through the pollen tube, it is known as stephanogamy. Entry of more than one pollen tube into the ovule is called as polyspermy. Polyspermy. Write complete sentences, both the points you note down. Fine. Completed all of you? Scribble down, scribble the things. Scribble, ಬೇಗ ಬರಿಬೇಕು. ನೀಟಾಗಿ ಬರಿತಾ ಕುತ್ಕೋಬೇಡಿ. ನಿಮಗಷ್ಟೇ ಅರ್ಥ ಆದ್ರೆ ಸಾಕು. I think most of them had practicals today. Practicals ಇತ್ತ ಇವತ್ತು?
Yes, ma'am.
I can understand by seeing your face. Okay. All of you had biology practicals or chemistry? Chemistry ಇತ್ತು ಅನ್ಸುತ್ತೆ ಕೆಲವು ಜನಕ್ಕೆ, ಹೌದಾ? Or physics? ಫಿಸಿಕ್ಸ್ ಇದ್ರೆ ತುಂಬಾ ಸುಸ್ತಾಗಿರಲ್ಲ, ಸೊ ಯು ವಿಲ್ ಬಿ ಫ್ರೆಶ್. ಬಯಾಲಜಿ ಅಂತ ಅಂದ್ರೆ ಕುತ್ಕೊಂಡು ಅದನ್ನೇ ಕೇಳಿ ಅದನ್ನೇ ಕಟ್ ಮಾಡಿ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಅಲ್ಲಿ ಕಾಣ್ತಿರಲ್ಲ, ಸ್ಲೈಡ್ ಬರ್ತಿರಲ್ಲ, ಮೇಡಮ್ ಬಿಡ್ತಿರಲ್ಲ. ಎಸ್. ಅಂಡ್ ಕೆಮಿಸ್ಟ್ರಿ ಅಂದ್ರೆ ಇಟ್ ಈಸ್ ಲೈಕ್ ರಿಸಲ್ಟ್ ಸಿಗೋವರೆಗೂ ಕಲರ್ ಚೇಂಜ್ ಆಕರ್ಸ್, ರಿಯಾಕ್ಷನ್ ಕಂಪ್ಲೀಟ್, ಟೇಕ್ ಡೌನ್ ದ ಇನ್ಫರೆನ್ಸ್, ಅಬ್ಸರ್ವೇಷನ್, ಸೈನ್, ಗೆಟ್ ಇಟ್ ಸೈನ್ ಅಂಡ್ ಗೋ. ಓಕೆ, ಕಂಪ್ಲೀಟೆಡ್?
Yes, ma'am.
Okay, let us proceed. Very important. Difference between egg cell ಮತ್ತೆ secondary nucleate. Egg cell ಮತ್ತೆ secondary nucleus. Secondary nucleus ಅಂದ್ರೆ ಇದು embryo sac ಅಂತ ಅನ್ಕೊಳ್ಳಿ. These 3 cells are antipodals. ಇದಕ್ಕೆ antipodal cells ಅಂತ ಅಂತೀವಿ. Then central cell ಅಲ್ಲಿ 2 polar nuclei ಇರುತ್ತೆ, which are haploid in nature. ಈ 2 ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈಗಳು ದೇ ವಿಲ್ ಫ್ಯೂಸ್ ಟು ಫಾರ್ಮ್ ಸೆಕೆಂಡರಿ ನ್ಯೂಕ್ಲಿಯಸ್. ಓಕೆ, ಸೆಕೆಂಡರಿ ನ್ಯೂಕ್ಲಿಯಸ್ ಅಂತ ಕರೀತಾರೆ. ಹಿಯರ್ ವಿ ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ಎಗ್ ಅಪ್ಯಾರಟಸ್. ಎಗ್ ಅಪ್ಯಾರಟಸ್ ಅಲ್ಲಿ ಏನೇನಿರುತ್ತೆ? 2 ಸಿನರ್ಜಿಡ್ ವಿ ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್, 1 egg ವಿ ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್. ರೈಟ್. ಸೊ ನಾವೀಗ ಏನ್ ನೋಡ್ತಾ ಇದೀವಿ? ಈ ಎಗ್ ಸೆಲ್ಲಿಗೂ ಮತ್ತೆ ಸೆಕೆಂಡರಿ ನ್ಯೂಕ್ಲಿಯಸ್, ವಾಟ್ ಈಸ್ ದ ಡಿಫರೆನ್ಸ್ ಬಿಟ್ವೀನ್ ದೀಸ್ ಟೂ ದಟ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ರೀಡ್. First point. Fatima, can you read first point from both the columns?
Egg cell. It is present near the micropyle inside the embryo sac. Secondary nucleus. It is present in the middle of the embryo sac.
Thank you, Fatima. So as she read, egg cell illirutte, micropyle end alli irutte, whereas secondary nucleus will be situated at the center. Yes. Diksha, can you read out second difference?
Egg cell. Generally, egg is surrounded by 2 synergic secondary nucleus. It is not surrounded.
Okay, thank you, Diksha. So as she read, the egg cell will be surrounded by 2 synergid. ಆದ್ರೆ ನೀವು ಸೆಕೆಂಡರಿ ನ್ಯೂಕ್ಲಿಯಸ್ ಅಲ್ಲಿ ಯಾವುದೇ ರೀತಿಯ ಸಿನರ್ಜಿಡ್ ಅಥವಾ ಸೆಲ್ಸ್ ಕಾಣ್ ಸಿಗೋದಿಲ್ಲ. Yes. Now, Manya, are you here? She's not here. Yeah, she's here. Okay. Third difference. Can you read third difference?
Ma'am, only single molecule is present to it. It is secondary nucleus. It is formed by fusion of 2 molecule nuclei.
Okay. So mute yourself. Thank you, Manya. Okay. Only single nucleus. Egg cell alli single nucleus will be present, whereas in a secondary nucleus it is formed by fusion of 2 polar nuclei. 2 fuse agi ಒಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಆಗಿರುತ್ತೆ. Next, Netravati, can you read out fourth difference?
It is haploid structure. It is diploid structure.
Very good. So egg cell ಏನಿರುತ್ತೆ? Egg cell will be haploid structure. It will be having single set of chromosome. Whereas central cell alli 2 polar nuclei, 2 haploid cells fuse to form diploid nucleus. So it will be diploid structure. Now, Jyoti, can you read fifth difference?
It is fertilized with 1 male gamete and to form diploid zygote. It fused with one male gamete and formed triplet primary endosperm nucleus.
Okay, thank you, Jyoti. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ? The egg cell will fuse with 1 male gamete to form 2n, that is a diploid zygote, whereas this secondary nucleus, which is already diploid, fuses with male gamete which is haploid. It forms triploid nucleus, third triple set, 3 set of chromosome. So this is regarding the difference between egg cell and the secondary nucleus. I hope you got it. ಅರ್ಥ ಆಯ್ತಾ ಎಲ್ಲರಿಗೂ?
Yes, ma'am.
Okay, please note it down. Note it down. If you completed, let me know. Okay. Mm. Once you completed, let me know. Jyoti. Is she present? Jyoti. ಯಾರ ಹೆಸರು ಹೇಳ್ತೀನಿ ಅವ್ರು ಸ್ವಲ್ಪ ಪ್ರೆಸೆಂಟ್ ಅಂತ ಹೇಳ್ಬಿಡಿ. Today's date is 20 na? Okay. Next. Anvita is not here. Muskan. Pooja illa. Santosh Meghana no. Nagalambika illa. Madhu illa. M Harshita.
Yes,
M
ma'am.
Harshita. Okay. Oh, 20. Completed all of you?
Yes, ma'am.
Okay. Next. Yes, Jyoti, M Harshita, Manya, all of you completed? Radhika, Mahalakshmi present?
Yes, ma'am.
Fatima,
Yes, ma'am.
Priyanka.
Yes, ma'am.
Okay. Manju and Sahana present. Shweta present. Lavanya absent. Pavan absent. Radhika absent. Okay. Fine. Thank you, girls. Next, we have development of endosperm. Triploid nucleus is formed. Why it is formed? To provide the nutrition. So endosperm which stored a food material. So endosperm, whatever the triploid nucleus is there, no guys, whatever, yeah, the triploid nucleus is formed, aden madutte, it provides food for the developing embryo. So primary endosperm, it will undergo repeated mitotic division and it forms mass of cell which is rich in stored food. It will be having stored food. So primary endosperm ididu endosperm agate fertilization admele. Okay. Next we have this food material is utilized by the embryo during early development. Food is present in the form of what? In the form of starch in the endosperm. So endosperm definition kelidre, it is a nutritive tissue as it provides nutrition, nourishment to the embryo. Adu ತಾಯಿ ತರ ಆ ಎಂಬ್ರಿಯೋನ ಮಗು ತರ ನೋಡ್ಕೊಳ್ಳುತ್ತೆ by providing the nutrients until embryo will be grown as a seed plant. ಅದು ಒಂದು ಚಿಕ್ಕ ಪ್ಲಾಂಟ್ ಆಗೋವರೆಗೂ endosperm will be nourishing it. In gymnosperm, endosperm is formed before fertilization and always haploid. Important for competitive ಅಂತ ಹೇಳಿದ್ರೆ ಕುತ್ಕೊಂಡು ಹಿಂಗ್ ಬರಿತಾ ಹೋಗ್ತೀರಲ್ವಾ. Okay. So in gymnosperm, endosperm is formed before fertilization. Gymnosperms ಅಲ್ಲಿ before fertilization endosperm form ಆಗಿರುತ್ತೆ. ಅದು ಯಾವ ಸೆಟ್ ಅಲ್ಲಿ, ಐ ಮೀನ್ ಯಾವ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಇರುತ್ತೆ? It will be always haploid in nature. Then again we have types of endosperms. Bantu nodu actual biology. So if you really love biology from the heart, then you will enjoy it. ನಾನು ಸ್ವಲ್ಪ ಡಾಕ್ಟರ್ ಆಗ್ಬೇಕಪ್ಪ ಎಲ್ರು ಅಂತಿದ್ದಾರೆ, ನಾನು ನೋಡಕ್ಕೂ ಸ್ವಲ್ಪ ಚೆನ್ನಾಗಿದ್ದೀನಿ, ಡಾಕ್ಟರ್ ಆದ್ರೆ ಇಟ್ ವಿಲ್ ಬಿ ಸೂಟೇಬಲ್ ಫಾರ್ ಮೈ ಫೇಸ್ ಅಂತ ಏನ್ ಅನ್ಕೊಂಡಿರ್ತೀರ ಅಲ್ವಾ. So for them, again, we have types. So it will become hectic. So enjoy biology, then you will enjoy whatever the class, then you can become a lovable doctor. So types of endosperms, Ali, three types we have. That is nuclear, cellular, halobial, NCH. NCH, not MTH. NCH. NCH andre N andre nuclear, C cellular, H halobial. So what are these? Let us discuss. Fine. Nuclear endosperm. As you can see, the endosperm undergoes nucleus division. Only nucleus of endosperm will be divided, and you can see free nuclei. Free nuclei ಕಾಣಿಸ್ತಾ ಇದೆ ನಿಮಗೆ ಹೌದಾ? Free nuclear floating. So such kind of endosperm is called as nuclear endosperm. Cellular endosperm andre nuclear division occurs where nuclear division is formed by cell wall formation. Nuclear division ಆದ ತಕ್ಷಣ cytokinesis ಆಗ್ತಿರತ್ತೆ. Cytokinesis ಅಂದ್ರೆ cytoplasm division ಆಗಿ each nucleus will be having its own cell. So it is called as cellular endosperm. Halobial endosperm ಅಂದ್ರೆ ಮಧ್ಯದಲ್ಲಿ at the center you can see nuclear endosperm, at the periphery ಅಂದ್ರೆ edge ಹತ್ರ you can see the cellular endosperm. So ಎರಡು ಸೇರಿ ಇರೋದ್ರಿಂದ it is called as halobial endosperm. It is called as halobial endosperm. Yes, Mahalakshmi, I took your attendance. Okay. Fine. So nuclear, cellular, halobial. Nuclear, only free nucleus can be visible. Cellular, nuclear division along with the cytokinesis or a cytoplasmic division. Halobial, centrally nuclear endosperm. Peripherally cellular endosperm. I hope you got it, guys. Yes. Okay. So this is regarding the sexual reproduction in plant. In the next session, we will be having embryo development. That takshana we will be solving the MCQs. Today you have some homework. I will just share the screen. Please wait for 2 minutes. Don't go anywhere because attendance and mark ಮಾಡಿ save ಮಾಡೋವರೆಗೂ you should be there. Okay. Neat. Fine. Okay. So let me share the screen. Okay. Fine. You can answer through the mic or you can answer through the chat. So the first question here, just check. Plant embryo develops from option A seed, option B fruit, option C zygote, option D flower. Plant embryo develops from. What is your answer, girls?
Zygote.
Zygote. Who answered this? May I know your name?
Apeksha.
Okay, thank you, Apeksha. So zygote is a right answer. Embryo of a flowering plant is always option A haploid, option B diploid, option C triploid. Okay, Lavanya, yes andre Netravati na? Fine, okay. Okay, so the answer is given by Jyoti, diploid. Answer B, diploid is a right answer. Next, plant embryo is a mass of option A cells, option B uncertain tissue, C collection of plant tissue, D miniature plant. Plant and embryo is a mass of what? Mass of cells anta helodalla. It is a mass of what? That embryo will become what next? It becomes a miniature plant or a plantling, right? So it is a mass of a miniature plant. Most of them will mark for cells. It is wrong. It is a mass of a future miniature plant. Next, stamen is a modification of option A leaf, Option B, microsporophyll. Option C, megasporophyll. Option D, shoot. ಇದನ್ನ ನಾನು ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಅಲ್ಲಿ ಹೇಳಿದ್ದೀನಿ, ಫಸ್ಟ್ ಅಥವಾ ಸೆಕೆಂಡ್ ಕ್ಲಾಸ್ ಅಲ್ಲಿ. ಹೂ ಅಟೆಂಡೆಡ್ ದೋಸ್ ವಿಲ್ ಬಿ ನೋಯಿಂಗ್. ಎಸ್ ಜ್ಯೋತಿ, ವಿಲ್ ಯು ಟ್ರೈ? ಸ್ಟೆಮನ್ ಇಸ್ ಅ ಮಾಡಿಫಿಕೇಶನ್ ಆಫ್. ಆಂಜಿಯೋಸ್ಪರ್ಮ್ಸ್ ಅಲ್ಲಿ ಸ್ಟೆಮನ್ಸ್ ಆಯ್ತು. ಜಿಮ್ನೋಸ್ಪರ್ಮ್ಸ್ ಅಲ್ಲಿ ಸ್ಟೆಮನ್ ಇಸ್ ಅ ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಪಾರ್ಟಿನ ಆಂಜಿಯೋಸ್ಪರ್ಮ್. ಜಿಮ್ನೋಸ್ಪರ್ಮ್ಸ್ ಅಲ್ಲಿ ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಪಾರ್ಟಿಗೆ ಏನಂತ ಕರಿತೀವಿ? ರಿಲೇಟೆಡ್. ಮೈಕ್ರೋಸ್ಪೋರೋಫಿಲ್. ಅಪೇಕ್ಷಾ ಟೋಲ್ಡ್ ದ ಆನ್ಸರ್. ಮೈಕ್ರೋಸ್ಪೋರೋಫಿಲ್ ಅನ್ನೋದು ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಪಾರ್ಟ್ ಇರುತ್ತೆ. ಎಲ್ಲಿ? ಜಿಮ್ನೋಸ್ಪರ್ಮ್ಸ್ ಅಲ್ಲಿ, which will be evolved, which will be changed as a stamen in angiosperms. Zygote is formed inside what? Stigma, style, female gametophyte, seed. Zygote.
Female gametophyte.
Diksha and Apeksha answered as female gametophyte. Right answer. So idrinda nimage artha agate. Most of you are answering, most of you are thinking. See how much content you know. You can cross verify yourself by this. ನಾನು ನಿಮಗೆ ಡಿಮೋಟಿವೇಟ್ ಮಾಡ್ತಾ ಇಲ್ಲ. ಐ ಆಮ್ ಟೆಲ್ಲಿಂಗ್ ಯು, ಯು ಮಿಸ್ಡ್ ದ ಸೆಷನ್ಸ್ ಅಂಡ್ ಯು ಮಿಸ್ಡ್, ಐ ಮೀನ್ ನಿಮಗೆ ಚಾನ್ಸು ಸಿಕ್ಕಿರಲ್ಲ ಕಾಲೇಜಲ್ಲಿ ಕರೆಕ್ಟ್ ಆಗಿ ಕಲಿಯಕ್ಕೆ, ಅಥವಾ ಅವತ್ತು ಫೋಕಸ್ ಮಾಡಿರಲ್ಲ, ಏನೋ ಆಗಿರತ್ತೆ. ಸೊ ದಟ್ ಈಸ್ ವೈ ಮೇಕ್ ಯೂಸ್ ಆಫ್ ಎವ್ರಿಥಿಂಗ್ ವಾಟ್ ಯು ಹ್ಯಾವ್. ಸುಮ್ನೆ ಕ್ಲಾಸ್ ಅನ್ನ ಬಿಟ್ಟು ಎಲ್ಲೋ ಹೋಗಿ ಯಾಕಪ್ಪ ಅದನ್ನೇ ಹೇಳ್ತಾರೆ ಅದನ್ನೇ ಕೇಳಿಸ್ಕೊಳ್ಳೋದು ಅಂತ ನೆಗ್ಲೆಕ್ಟ್ ಮಾಡಬೇಡಿ. ಓಕೆ, ಸಿಕ್ಸ್ತ್ ಕ್ವಶನ್. ಮೈಕ್ರೋಸ್ಪೋರ್ ಈಸ್ ವಾಟ್? Male gamete, first cell of male gametophyte, last cell of male gametophyte, diploid cell. ಏನಿದೆ ಅದು?
Diploid cell.
Gamete.
Diploid cell.
Two of them answered as diploid cell. One answer was different. Who answered this and what was your answer?
A male gamete.
Okay,
And
Apeksha
diploid
answered
cell.
as, okay, Apeksha answered as first cell, sorry, male gamete, and diploid cell is answered by Jyoti and someone. Okay, here a microspore is a male gamete alike. Even though it is a male gamete ಆದ್ರೂ answer ಗೆ right, I mean you will get the marks. But the answer here they expect is first cell of male gametophyte. ಅದು male gametophyte ಇನ ಒಂದನೇ ಸೆಲ್ಲು, first cell. An anther consists of, anther ಅಲ್ಲಿ how many microsporangium you will be having? Anther
4 microsporangium. 4.
Okay, very good. Shweta answered it. 4 microsporangium will be present. So stamen iratte, anther iratte. So when we take a transverse section, you will get the section like this, which will be having 4 microsporangium, 4 bag-like structure. Okay, Mahalakshmi, good. Cells of nucellus are always, nucellus in a cells, haploid, triploid, diploid, enucleated. Nucellus.
Diploid.
Diploid. Okay, right. Diploid is a right answer. The embryo sac is produced from, embryo sac or a female gametophyte to ಯಾವುದರಿಂದ produce ಆಗತ್ತೆ? Option A, microspore. B, zygote. C, egg cell. D, megaspore. Concentrate and then answer. Yes, Jyoti? Shweta?
Megaspore.
Very good. Megaspore. The embryo sac is produced from megaspore. Now, most of them we will be voting for egg cell. Egg cell in the megaspore in the embryo sac agate. Embryo sac ali egg iratte. An egg apparatus contains, egg apparatus al ಏನೇನ್ ಇರುತ್ತೆ? Option
An egg plus 2 synergids.
Okay, good, Shweta. An egg plus 2 synergid is the right answer. Very good. Great going, girls. So please focus on all the questions. Please focus on the content. Don't get demotivated. ನಾನು ಏನು ಆನ್ಸರ್ ಮಾಡ್ಲಿಲ್ಲ ಅಥವಾ ನಾನು ಟೈಪ್ ಮಾಡಕ್ ಆಗ್ಲಿಲ್ಲ ಅಂದ್ರೆ ಸ್ವಲ್ಪ ಜನ ಓವರ್ ಕಾನ್ಫಿಡೆನ್ಸ್ ಅಲ್ಲೂ ನಾನು ಯಾಕೆ ಹೇಳಿ ಇಷ್ಟು ಈಸಿ ಕ್ವಶನ್ಸ್ ಅಂತ ಇರಬಹುದು. ಥಮ್ಸ್ ಅಪ್ ಫಾರ್ ಯುವರ್ ಓವರ್ ಕಾನ್ಫಿಡೆನ್ಸ್, ಬಟ್ ಡೋಂಟ್ ಕಂಟಿನ್ಯೂ ಇಟ್. Then who has answered it well and good? Who has tried it well and good? ಲಿಟ್ರಲಿ ನಿಮಗ್ ಗೊತ್ತಿರತ್ತೆ ನೀವು ಆನ್ಸರ್ ಮಾಡಿದ್ರೋ ಇಲ್ವೋ, ನಿಮ್ದು ಟ್ರೂತ್ ಇದೆನೋ ಇಲ್ವೋ, ಆನೆಸ್ಟ್ ಆಗಿದ್ದೀರೋ ಇಲ್ವೋ ಅಂತ. ಇಫ್ ಯು ಫೀಲ್ ಯು ಆರ್ ನಾಟ್ ಪ್ರಿಪೇರ್ಡ್ ವೆಲ್, ನೌ ಯು ಹ್ಯಾವ್ ಅ ಲಾಟ್ ಆಫ್ ಟೈಮ್. ದಿಸ್ ಈಸ್ ಅ ಅಲಾರಾಮ್. ಕಾನ್ಸಂಟ್ರೇಟ್ ಆನ್ ಇಟ್. ಎಲ್ಲಿ ತಪ್ಪು ಮಾಡ್ತಿದ್ದೀರಾ ಅನ್ನೋದನ್ನ ನೆನ್ಸ್ಕೊಂಡು start doing it and start practicing it. I will be always with you, girls. Let us end up the session here. Have a nice day. Bye bye.